Hi there. I hope you can hear me okay and see my screen. Welcome and thank you for joining us for this webinar on the use of chlorine dioxide in bulb dipping. I'm Natalie Key, Knowledge Exchange Manager for Horticulture at AHDB. And before we kick off, just a couple of housekeeping elements to go through with you and also what to expect from the webinar. So for information throughout the webinar, the audience is muted, but you can submit questions through the question box on the right-hand side of your screen. Any questions that don't get answered during the webinar, we'll try to follow up with after the webinar. Similarly, if you think of something you want to ask once we're finished, please do email me on natalie.key@ahdb.org.uk. I will read questions aloud during the webinar to the presenters. If there are any issues during the webinar that you can't flag through the questions box, please also email me and I'll keep an eye on my inbox. The webinar will be recorded and made available on the AHDB events archive after the event if you want to catch up on anything. And there are basis and Enroso points available for attendees, so please complete the forms that you can download from the handouts box on the right hand side and return them to Maya, whose email is also listed on the form. So we're going to start off with Rob from Warwick Crop Centre, who'll provide a bit of background on the work and talk about his work testing the efficacy of chlorine dioxide in controlling levels of fusarium in bulb water treatment, and also the examination of the effects of this treatment on bulb and plant health. We'll then pass over to Ewan Cameron from Scott Mass, who will discuss how the chlorine dioxide application works in this context and how growers could use this technology in practice. Finally, I'll wrap up with some comments and information about the growers who were involved with this trial work. We'll do our best to keep to time, should be finished around 1:30, 1:35. So I will now pass over to Rob to start us off.
Good morning, everyone. Hopefully you can see my screen in front of you. If you can't, send me a text.
We can see you on your screen.
Excellent. So I'm sure there are at least 9 of you present. At least 8 of you have probably seen some of this before. For that, I apologize, but I'll try and make it brief and informative as we go through and address the question, does chlorine dioxide reduce the level of fusarium in Narcissus? So this project, BOF 77, was commissioned just over 4 years ago and finished this spring. So this is the first opportunity we've had to report the results in full. Now, I know you're probably all aware of most of the background, but I'm quickly going to run through it just to refresh your memories. So we're starting back in the times of formaldehyde, when that was freely available, and we used that in pretty much all of our hot water treatment systems. This was, of course, withdrawn in 2008, and we then had to seek alternatives. A number of candidates were tried back at the time, and FAM30 was identified as being the best replacement, but even so, there were some concerns about its efficacy as the active ingredient, which is iodine, rapidly dissipated in hot water systems. And that is an issue that we have with many of the biocides, in that these biocides, and to some extent the fungicides as well, are all designed for use at ambient temperature, and we're putting them in an aqueous hot environment, and that is not their preferred environment, and we lose efficacy at every point. But the search continued beyond FAM30, and one of the first alternatives that we looked at was chlorine dioxide, as this had been used and investigated in the States back in the early 2000s. And in Project BOF 70, Gordon Hanks investigated the use of chlorine dioxide in a UK framework, and found that there were no negative results of stem length, development rate, or incidence of disease if chlorine dioxide was used in hot water systems. They did take the experimental through to actually putting bulbs down in the field, and there were some concerns raised at the time over reduced yield where chlorine dioxide had been used. And for that reason, they decided that they couldn't recommend chlorine dioxide and that they suggested that everybody continue using FAM30. And that is really the position that we had for quite a while until the commissioning of this particular project. And what this project tried to do was to further that work on chlorine dioxide and others to see if new ingredients or new processes could bring about an improved performance. And that's where we stepped in. So BOF 77 proposed a three-stage approach to testing chlorine dioxide and a few other candidates, but we're not going to discuss those at this stage, and to see if we could extend the work that Gordon Hanks had done back in BOF 70. And to this end, we envisaged a three-stage test. And the first one was a purely laboratory test to investigate the efficacy of chlorine dioxide just to control spores. And we assumed that if that was successful, we would move on to a small-scale tank test where we would actually bring in bulbs as well as fusarium spores to see if we could understand the combination in hot water systems. And assuming that that was successful, we would then move on to a commercial test using on-farm full-size tanks. So the first stage was the laboratory tests. And we were starting from a position of knowing a reasonable amount, in that BOF 77 had showed that chlorine dioxide is and was an effective biocide. So we knew that it could be used effectively against pathogens, particularly fusarium, as it had been used in other industries, and it's used throughout the medical and food industries. But when Gordon was running these particular experiments, he came up against a problem that we also experienced, in that the very nature of hot water systems, we've got hot water, it's dirty, reduces the efficacy of all these biocides, and chlorine dioxide very much fell into that category. So we were both testing the candidate biocides, but we were also trying to think through how we were going to deal with this bioload, this mixture of soil particles and sludge that builds up in treatment tanks over the dipping season. And bioload is predominantly soil particles. So it's soil particles that are being carried in, adhering to the bulbs, and as soon as the bulbs get into the hot water, the particles leave the bulb scales and then just get circulated throughout the water system. And our first step was to do laboratory testing at selected concentrations of chlorine dioxide on both clean and dirty water systems loaded with fusarium spores. And it was really at this stage to see what was the effect of concentration and what was the effect of clean and dirty water. And at this stage, we were using a liquid combination to generate our chlorine dioxide. And the pictures on the right-hand side show you just how much variation it's possible to get in tank waters. Now, all these samples were collected from commercial growers a couple of years ago as part of this trial, and they show you just how dirty water gets. So some tanks, some growers are quite good at keeping everything relatively clean. It's not possible to keep it completely clean, but in other systems, if there's more soil involved or different soil types, the water can get dirty quite quickly. So the first laboratory test was a dose response test. And in it, we were looking at 5 concentrations of chlorine dioxide, so 1, 2.5, 5, 7.5, and 10 parts per million chlorine dioxide. We were looking at 5 different durations of treatment, going from a very brief 5 minutes up to the full 3 hours of a standard commercial treatment. And this figure shows the results across those 2 sets of criteria. The values are spores per milliliter. So the spores that we generated, these are the ones that were added to the mix, and it's shown as a logarithmic scale. So 0 is obviously 0, but 1 times 10 to the 7 at the top is actually 10 million. So when you get your heads around the scale, it starts to become apparent that 1 and 2 parts per million is not overly effective. 1 part per million actually doesn't do a huge amount in comparison to the control that's at the far side. So we're still looking at about 10 million spores per milliliter. If you increase the concentration to 2.5 parts per million, it shows a halving, but it's actually more than a halving because it's taking it down from 10 million down to 10,000. And as you increase the concentration through 5, 7.5, and 10 parts per million, you can see that the concentrations of spores that survive the chlorine dioxide actually fall down to sort of 1 to the 2 and 1 to the 1, so somewhere between 10 and 100 spores per milliliter from a starting position of 10 million. And in terms of this sort of trial, that's pretty much complete control. So this gave us the evidence to suggest that chlorine dioxide is actually a very effective biocide when it's done under controlled conditions. Now, there's no bulbs at this stage, but it's just an initial proof of concept to say that chlorine dioxide is a good biocide. And our conclusions were that concentrations at 1 and 2.5 parts per million did not prove effective control, but that 5 parts per million and above did. But it also says that actually the duration of the treatment was not significant under these conditions. And these are lab conditions, but it's a useful piece of information. At the same time, we were looking at both clean and dirty water systems. And for this, we used 7.5 and 10 parts per million chlorine dioxide. And if you look at the figure, it's very apparent that clean systems provide actually very good control. So down from my 10 million spores per milliliter down to almost 10, possibly 100 spores per milliliter, showing that chlorine dioxide is a very effective biocide in clean water conditions. Unfortunately, it's not a good biocide in dirty water conditions. And that is something that we expected, but it's nice to show it being proved in these results. So the conclusions at this stage were that clean water results confirmed that chlorine dioxide is an effective biocide at 7.5, 10 parts per million, but that efficacy was compromised in dirty water, where, to be honest, there was little difference between treated and untreated controls. So that was a disappointing, if not unexpected, result. Buoyed by this, we moved on to sort of small scale tank tests. And this is a similar approach, but just upscaled. So we were going to look at the efficacy of chlorine dioxide in hot water treatment to control pathogens on the incidence of disease is in bulbs. So this was the first time that we introduced bulbs into the system. And again, we ran both clean and dirty water experiments. Fusarium spores and bulbs were added to tanks and mixed, and tanks were maintained at the industry standard of 44.4 for 180 minutes. This time around, we used tablets to generate the chlorine dioxide, but used the same concentrations. But we also played around a little bit with the delivery of the chlorine dioxide, in that we either used a single burst right at the start of the experiment, or we maintained the concentration throughout the experiment. And we took water samples at 5 and 180 minutes to see what the results were. So this is the results of the small scale tank tests. And it takes a little while just to get your head around this. So the blue bars indicate the level of spores, as in colony forming units, across the treatment types. So treatment 2, which was clean water, treatment 4, which was 5 parts per million continuous, treatment 5, which is 5 parts per million single, etc., etc. So 10 parts per million as well. And what the results showed was that if you tested at 5 minutes, actually there was very little reduction in terms of spores. So the blue bars are 5 minutes, and whatever treatment you had, there was pretty much no reduction in spore numbers. What it does display is the fact that we did have spores there. So that was just an effective control. The green bars show you the level of spores at 180 minutes. And these show an almost complete control across all the different treatments. So the only time there wasn't complete control was in T2, where we didn't actually add any chlorine dioxide. All the other treatments where we had chlorine dioxide, no spores survived after 180 minutes. And that is the level of control that you're looking for in this sort of experiment. If you quickly return to T2 and look at the difference between 5 minutes and 180 minutes, the slight improvement for 180 minutes is the effect of heat. So that is what 3 hours at 44 degrees does to spores. It will probably control about a third of the spores that are in the tank. So conclusions from this particular stage were that 5 minutes, the samples confirmed the presence of fusarium spores in our system, but actually really no control. At 180 minutes, the control treatment demonstrated that heat alone only reduced rather than controlled spores, but that at 180 minutes, all of the treatments provided effective control of spores. And this gave us confidence to move into the next stage. But we also looked at bulbs that came out of these tanks. So post-dipping, all the bulbs that we were looking at were incubated at 25 degrees centigrade to accelerate development of any disease. And at the end of 28 days, we dissected all the bulbs and scored them on infection on a 10-point scale. And that 10-point scale is shown on the right side of the screen. Now, you don't need to take it all in, but really 0 and 1 is very, very low infection. 8, 9, and 10, it's not good. Everything is infected. And the results of the bulb assessments are shown in this figure. And I think there are 2 main takeaway stories. The first one is look at T1. So this T1 is only just clean water. And in that, there was no infection, showing one, that the stock of bulbs we had was very clean, but equally that none of the bulbs became infected. T2 is the other extreme. So T2 is water plus spores, but no chlorine dioxide, which shows that 90% of the bulbs became infected. And this confirms the strength of the treatment of the spores that are in the water, and demonstrates that with no control, pretty much all of your bulbs would have become infected. And once you get beyond that, looking at treatments T3 through T10, although there is some variation, and some treatments, particularly T7, perform better than others, what it is demonstrating that all of the treatments provided some control, and they were all better than doing nothing. And that is the sort of confidence that we wanted to go forward into the main tank tests. And the conclusions from this stage were that in the absence of chlorine dioxide, only 10% of bulbs were not infected. But controlling clean water was obviously better than dirty water, and that 10 parts per million was more effective than 5 parts per million. Although, to be honest, there isn't a lot in it. And economically, 5 parts per million is always going to be better than 10 parts per million, as Ewan will demonstrate later. So having done the laboratory and the small scale tank testing, we moved on to test at a commercial scale. This was done at Jack Buck's Farms up near Spalding during the summer of 2018. And Ewan from ScotMass, who you'll hear from in a minute or two, installed a semi-automated dosing system, and we tested that over 3 weeks during the bulb dipping season, just really to fine-tune it so that we could get the right amount of chlorine dioxide going into the tanks. And at the end of the 3 weeks, all the treated bulbs were replanted alongside the farm's normally treated stocks. And assessments were then carried out in 19 and 20 to monitor plant growth and flower quality. But Ewan will tell you much more about this in his presentation. So we went back in spring 2019 to assess the first year flowers, and that was done on the 20th of March. This was really just a look-see. So we took just 20 stems from each batch, and really we're just looking at stem length and any physiological damage. And basically the results showed there was no significant differences between the batches. And we looked at 3 batches. Batches 1 and 2 were just evaluation, where Ewan and I were playing around with the concentrations and just trying to get it right. But even those, and we dismissed them as not being that great, when you look at the flowers, it didn't actually make a huge amount of difference. The third batch, which is where we considered we'd got the optimum amount of chlorine dioxide, was pretty much the same. There were no obvious differences between any of them and the control. So at this stage, we're very happy. We're not really looking for improvements. We were really looking for detrimental effects. So I went back this spring to do an assessment on the second year flowers, which is on the 23rd of March, which I'm sure we all remember as being the first day of lockdown. So I was a bit of a naughty boy and strolled up to Lincolnshire. And we did a full assessment on 300 stems from batch 3 and the control. And the results showed that the chlorine dioxide treated bulbs performed better than the control across all 3 measured parameters. Now, I wasn't really looking for better. I was actually looking for about the same. It's nice that it's better, but don't read too much into that. What I wanted to check was that chlorine dioxide was not detrimental. And the results showed that there was no difference between the chlorine dioxide and the normal farm method. So that showed that chlorine dioxide in this instance was actually perfectly safe and had no detrimental impact. And that to me is the main outcome of this project. We know we treated it, it's been planted, the flowers are being collected, and there was no difference. That to me is a very successful result. So just to conclude from this short presentation. So chlorine dioxide is an effective biocide for use in hot water treatment of Narcissus bulbs. We've shown that at lab scale, at small tank scale, and at commercial scale. And these results confirm its efficacy against fusarium spores on one side and the development of basal rot as well. So we're happy that it controls spores in the tank, and we're happy that that positive benefit is seen through to the development of basal rot. It's not all good news, and we always have to remember that high bioload can reduce its efficacy. So almost my plea to growers is to keep the system as clean as possible. And the other thing that came out of this particular bit was the development of an automated dosing system. And that overcame many of the difficulties that other researchers had experienced in earlier trials, where single dosing was a far more common approach. And I think it's that development of that automated system that constantly monitors the concentration of chlorine dioxide that will give confidence both to me as a researcher and to the growers as well. Thank you very much. I'm not sure if we're taking questions now or later, but I'll let Natalie dissolve that one.
Yep, we've got a little bit of time for questions. If anyone wants to ask Rob, just submit them via the questions box on the right-hand side. I've got a couple for you. A question about the small tank tests. What was the source of the water you were using? I'm not sure whether you mentioned that or not.
So the water was just water drawn from our own well or our borehole at Wellesbourne, and it was either clean or it was dirty, and the dirty was a mixture of soil particles and bulb scales that we mixed up to make a dirty tank.
Okay. So you conclude that increasing the concentration of chlorine dioxide in dirty water is not effective at overcoming the reduced efficacy that you saw in dirty water in terms of using fusarium spores?
So the dirty water scenario was the one that we tested using the small scale tanks, and for that we found that the efficacy was certainly reduced in dirty water, and that is one of the reasons why Ewan and Scottmas were so welcome into this piece of research, as the development of their automated dosing system has overcome the bioload. But I'd say Ewan will talk more about this in his presentation.
Okay, thank you. Did you test this? Did you test chlorine dioxide treatment in cold water situations as well?
So the initial laboratory tests were done in cold water. The ones that we used to select the candidate biocides and to test their efficacy against fusarium spores, that work was done in cold water, and the small scale and obviously the farm scale tests were done at 44.4.
Okay. A couple of questions. Well, what are the next steps, or do you think further trials are required, or would you say that the results are fairly conclusive in terms of, you know, potential benefits of using this in a commercial setting?
I think there's two parts to that question. As a researcher, I'd always say that more trials are required. I think that's inevitable. Yes, this is just the result of a single project done on a single commercial site. Now, I see no reason why the results aren't transferable, and I'm very confident that they will be. And I know that there are still some ongoing farm tests being done on a commercial basis. But having said that, I am very happy and confident with the results. And I think that further testing would probably only just confirm what we've already seen. But if you want to give me more money, I'm happy to spend it.
Noted. Final question, how important is the pH level of the water?
So tanks, of course, are notoriously acidic. We didn't experience any issues with pH, but I think Ewan is probably better equipped to answer that at the end of his presentation.
Okay, so perhaps well, unless Ewan wants to dip in now, or should we come back to that one?
Yeah, it's not a problem for me to answer that directly at the moment. Chlorine dioxide, one of its main benefits is that it's not affected in any way by pH. If anything, the more acidic that the water is, the better the conversion of stabilized chlorine dioxide into active chlorine dioxide is. So one could say that in actual fact the acidity is a benefit, if anything, But technically speaking, according to academic research, chlorine dioxide is not affected by pH.
Thank you, Ewan. Okay, I think that's it for questions for you, Rob, for now. But if there are more questions that people have, please submit them and we can come back to them at the end as well. Thank you, Rob.
I'll be here.
Pass over to Ewan now.
All right, do we have my screen?
We do, yeah.
Just have to maybe take my picture out of it because I can't see it myself. There we are. Right, we've got to start. Okay, well, following on from the Rob's, we've obviously got exactly the same angle that we were looking at, and that was to try and introduce a suitable automated system to try and control the fusarium. We'll just have a quick look.
Ewan, would you mind putting it in presentation mode, please, if that's all right?
Mine should be in showing full screen.
We can just see it in your in the PowerPoint window at the moment.
I'm not sure I've got ability on this screen here to do that. How do I do that?
At the bottom, there's a little symbol by the right at the bottom along the bar. There's a little symbol next to the focus in element. It's just there. It's like a little box with on a stand. Can you see that? Just right at the bottom.
I don't have that on mine.
Okay, well that's okay. We'll just continue. That's fine.
Apologies for that, but I don't seem to have that symbol anywhere on this. Oh, my apologies. It was working earlier. So just a quick introduction basically to Scott Mass. We're obviously a chlorine dioxide specialist, and we've been involved in the business for approximately 30 years, and we're probably the first European manufacturer to gain recognition and regulatory approval for the use of our products in drinking water for human consumption. So from the basis of it being a sound, safe and environmentally friendly product, you can obviously establish from the fact that it's used in human water that we tick all the boxes on that. We're probably, although there are, as you'll be aware, most of you, that there are a number of chlorine dioxide companies, which you can obviously Google and find out their strengths, and I'm sure you'll find that there are some companies there with some very good products along with ourselves. However, we are probably one of only a couple, if not the only company in chlorine dioxide where we actually manufacture the product ourselves in terms of the precursor chemicals, and we also make our own equipment in-house, and we also have technical specialists in each of the application areas that we deal with. So it is very much a one-stop shop, so to speak, and all our equipment and everything which we manufacture here is all not only designed by ourselves, but it's also tested up to RAS approval, which is the Water Research Council, and anything which comes into contact with drinking water must be RAS approved, and we are probably one of the only ones in Europe at the moment that have the full specification of regulatory approvals for this to be allowed. So there are another other companies out there which do have equipment, but one of the strengths of Scott Mass is that every single one of our components is actually approved. So we take on board all the background from drinking water, and we'll show you what we've done in terms of bringing that into play for fusarium control. We manufacture a number of pieces of equipment. All of it, generally speaking, is bespoke and it's designed to fit into the application areas such as cooling towers, municipal drinking water, odor control, and livestock and hospitals, etc. We have a number of units, varying sizes, so don't get frightened. This is not the sort of unit that we would be envisaging putting out on farms, but this in actual fact is the world's largest generator that's been built by Scott Mass, and this is now in place in Qatar, and it will actually be supplying all of the water for that country, and its main aim was to ensure that water quality was extremely safe for the up-and-coming Football World Cup. So our extensive knowledge has now been put to sort of worldwide use. So we have strengths in that area. Chlorine dioxide is our main and only, perhaps only product that we really specialize in and manufacture. And we are now at the point where we have now commissioned several months back our precursor production plant, which means we're probably one of only 5 in the world which can produce the chloride solutions which are used in making up the chlorine dioxide. So we're getting stronger the more we take on control of the full manufacturing process of our products. As we said earlier, it's been approved as a drinking water for human consumption, and the breakdown components are salt, so it means basically it's environmentally friendly as you can get, and it is basically a broad spectrum biocide, so we can actually control not just spores but also bacteria, viruses, and a general very quick kill because of its what's called free radical chemistry. It's very low in corrosion, and it's far more effective than chlorine at the same dose rates. As we can see here, the chlorine dioxide, which is actually available to do the kill, is affected by the bioloading, but not to the same effect as it would be had we been using just, for example, chlorine. There's far more of the chlorine dioxide available because it really is actually looking to rob electrons, and it's very selective in the way it does that, and it doesn't actually combine as it would do if it was with chlorine. So there's far more available biocidal effect than there would be again with other types of products such as potassiums or hydrogen peroxides or peracetic acids, etc., which other people have tried in the past. That just shows you there just exactly the differences, and I've compared it with chlorine because it's just more of a, I know it's not a fungicide as such, but it just gives you the overall impression of just how much of a biocide is actually available to do the work that we're trying to do. And this becomes very important when we're actually looking at the soil buildup and the bioloading which is inside these tanks, which we can show later on. Because of the effects which chlorine dioxide has, we can see here the number, and I've just selected these out, so it does say that it's not an exhaustive list. It's just basically to give you the impressions of what chlorine dioxide is actually effectively used for in controlling. You can see there it's mildew, botrytis, rust, and various other things. Then we go into pathogens. There you can see fusarium is listed. Nematodes is a very interesting one as well. We've been doing some work on that one there, which might be of interest, and we can maybe touch on that just right at the end, but it's obviously not part of today's concentration. Salmonella, Listeria, Pseudomonas, and other bacteria, which are obviously very useful in food processing areas. We see there soft fruits, horticulture, orchards, root crops, even arable, which we've gone into investigating at the start of last year, and we're continuing that on this year, and we're actually seeing some very good results in controlling various infections in arable crops. Irrigation water, dosed water, which is obviously greenhouses, etc., which we're very research heavy at the moment. We've got all the items. It's non-toxic, non-corrosive. There's no residual, and that's a very important one for harvesting, so that you don't have any withdrawal period. So you can actually spray in the morning and harvest in the afternoon. So people such as grape growers and soft fruit growers, this is a very useful aspect of chlorine dioxide. No resistancy. Now this again is a very, very useful aspect of chlorine dioxide, because it does mean that year on year you will have the reliability that chlorine dioxide will continue to do the job for you in 10 years' time as it's going to do this year. There are a number, obviously, of products which are on the market which slowly get more and more resistances built up, and they become less and less effective year on year. That doesn't happen with chlorine dioxide. Because of its ability, its solubility, it is very even distribution within your system, so we don't get pockets of high concentrations or areas in tanks and recirculating systems where we're going to get no contact whatsoever. So it's a very nice, evenly distributed, available product. We are reducing maintenance costs. That really is relevant for things like corrosion, and that also applies to plastics and rubbers as well as it does metal. Most people assume that corrosion is basically only in metals, but in actual fact there are other components which are very susceptible to embrittlement, which is another form of corrosion. Seed treatments, etc., which we can do. We'll hop over that though. Misting systems, again, removing all the biofilms and things which are inside pipelines is very, very important, so we don't have cross-contamination. Pre and post-harvest, so we'll look at them. Now, today, really, we're talking about fusarium, so obviously we're looking at how we've gone about looking at the control mechanisms, the equipment we used, and problems encountered, and how we've gone around trying to resolve them. These are some of the tanks which we've been looking at. I think you're all probably familiar with them. We've got open top tanks, we've got side entry tanks, which we were using both at Jackbucks and at Greenyards. So you can see there the difference, and you can see in here the water quality varying from the start of the season and the tar that we ended up with at the end of the season. And this basically goes back to the bioloading, which Rob was pointing out, and the effects of chlorine dioxide on something like that, and we'll talk a little bit about how we have managed to overcome that. The automated system, this is in actual fact just a test rig, so this was not the sort of commercially proposed type unit, but it served our purposes. We've got two separate dosing pumps here which are connected to the two precursor chemicals. One is an acid, as mentioned, and this is the chlorine dioxide. Now, the reason why we have two chemicals here is that the chlorine dioxide, when it's stabilized, has a buffer, and that makes it easy for us to transport without any risks or issues or health and safety problems. But at that particular stage, chlorine dioxide is pretty ineffective, and in fact, it would be lucky if it's considered biocidal, never mind biostatic, never mind biocidal. So what we have to do is we have to remove those buffers which are inside this solution, and that's what we require the acid for. So the acid and the chlorine dioxide mix in the chamber, which is up here, and we'll have a closer look at it in a minute. But this chamber here takes in these two chemicals. There's a resin-coated and etched glass, basically marble type balls, which are kept in here to increase the surface area. That gives us a complete reaction, and then we have two valves at the top here in this particular instance, because we were feeding onto two tanks, one on the left and one you can just see here on the right. So again, this is just a close-up now of just the system here, where we've got two digital dosing pumps, because obviously we're setting it down into parts per million, which means we're using mils of chemical per ton of water. So they need to be digitally controlled. And we have an analyzer here, which records or gives us a visual detail on what exactly the part per million concentration is in the tank. So this is fitted up into there's a probe here, and you'll see that there's a water feed that's bringing water from the tank through the probe. It's analyzed, and the figure is up here. Now that just basically allowed me during the trial to monitor what effect the chlorine dioxide was having. And the way we do that is by a residual value. So if there's a high loading, the residual value of chlorine dioxide will drop. therefore we have the ability then to alter the pumps to boost it back up again. So we were manually trying to keep a steady residual value in the tank. Again, you can see here the difference here. This is when we took water from the tank, we added 20 parts per million of chlorine dioxide, and we saw an immediate change in the color. So we knew that the chlorine dioxide was being effective in this particular case, and we were able to look at the loading afterwards, which was excellent, although the color was still very much a dirty color. These are just some of the bulbs which were post-treatment cooling off. And you can see here now how the bulbs were loaded in. So there's very, very compact scenario inside these tanks. So one of the key areas that we needed to determine was whether or not the chlorine dioxide was going to evenly distribute itself so that we got through the whole of these boxes. And you can see here from this photograph here just exactly how dense those bulbs are. So we needed to be assured that the chlorine dioxide wasn't just going to circulate around the top surface of these bulbs or just skirt around the side. We needed to make sure that we were getting good results right down into the center of these loaded boxes. And that is part of what we looked at, and we were quite successful, probably due to the solubility of the chlorine dioxide. It wasn't glooping up and actually getting stuck. It was being evenly distributed throughout the duration of the 180 minutes of the cooking process. So this is now taking us on to the previous trial units to what we've now designed as what would be now a commercially viable unit for this particular type of project. What we've done here is we've moved away from having a manual monitoring system to now automating it. So it has exactly the same principle, but this now will automate with and talks down to these pumps here, and whenever the residual level falls below what we've set it at as a minimum requirement, it will automatically bring the dose rates back up again. So we now have a computerized version of me hand altering the dosing pump. So this is now all set into a computer program. We've now also moved the activation chamber down into here. So we have a variable size activation chamber depending on exactly what size of tanks you've got. And you can see here now there's a plumb fitting. So this can now be moved on to any site and just automatically clipped in. This gets around a little bit of us having to make a bespoke system, which would cause probably complications throughout the industry of us having to make a different unit for every farm. We now basically would just have to probably at best alter maybe the pipe size, but everything now is in a relatively compact unit. It's just a plug and play system now, and we can pre-program as much of the details of what we want to do in terms of dosing into that unit, and you can just walk away and leave it and it'll get on with the job. So yeah, and you can see here we have added in also the ability now to dose more than one tank from the same dosing unit. So instead of us having up here, as we saw before, where we've got a tank on the left and a tank on the right, and we have to manually control which way it goes, we can now do automatically with these solenoids. And again, you can pre-program that into your computer on this one here. We can tell it we want to run for X number of hours onto tank on the left, and then we can switch it automatically either on a timer or on a solenoid to go into, so you wouldn't have to have a situation of having more than one dosing unit. So for those of you who have more than one tank, then obviously you would benefit from that probably more than people who just have a single tank. Again, this is probably more an advantage for people with glass houses and people who want to add nutrients into the actual growing period. So this is not quite as relevant, but it does, with these multiple dosing heads, we can actually put in other treatments into the tank if you so wish. So there is As you can see here, just we can add anything up to probably 16 different pumps, which means you will have full control over anything you would want to put into it. But as I say, that's probably not quite as relevant for this particular session. So these are just for security and safety. We have alternate methods, as we've highlighted, for measuring the chlorine dioxide. We have simple test strips here, which are very, very cheap. But obviously, this is only of any use if you're actually manually running your system and you need some control. These other ones here are more sophisticated versions, but still they're a manual check, as is this one here, which is just a wall-mounted pH and chlorine dioxide residual probe. This is the full monitoring system, which is in line. So you could have this in actual fact sitting either in an office, so you could have somebody who wanted to not necessarily be in and out looking at the equipment out on site. You could actually monitor this over the internet, and this dosing system can actually sit in a quality control area, or in, as it would be, your production manager's office or such like. So we've advanced it up so that we can actually program these units here, and it will automatically send the signals to your pumps, and that gives you an absolute constant flow of chlorine dioxide at the right levels. And I think that from Rob's work, you saw that there was very little of a difference between 5 parts per million and 10 parts per million, and that really comes down to, as he mentioned, economics. And the way chlorine dioxide works is it's mutually exclusive with bacteria, or in this particular case with the fusarium. So if you start having an increasing concentration of parts per million, it's almost a waste, because chlorine dioxide, when it's killed everything, it just stays stable. It doesn't actually go anywhere, it just stays in the system. So 5 parts per million is probably, on the tests we did in the last year or in the second year of this trial, anywhere between 5 and 7 or 8 parts per million was giving us very, very good results in terms of the stability of it. 10 parts per million is probably overkill, and therefore you can actually start saving money by bringing that dose rate down, and it can be done automatically. What you do have to watch out for, obviously, is that occasionally there could be higher demand in certain batches because they're not as clean as the batch before, and that means we don't want to preset the pumps. We want to allow the residual monitoring to take control over what it thinks it needs to put into the tank. So it allows you that flexibility to be able to either do it automatically or semi-automatically. For individual tanks and people who don't want all the sort of monitoring facilities and the added expense, we've now produced a much smaller unit. Obviously, it's not as robust as the larger unit, but certainly it has capabilities similar to the larger unit, but without the monitoring facilities. So we are looking at options which obviously is designed around it being cost effective for the growers, so we can actually make units that are budget sensitive, shall we say. I don't want to go into too many of these things, but this is just to show you the flexibility of the dosing systems. We can control many different aspects of it at a touch of a button, basically, and graphs can be produced which show you what net effect your chlorine dioxide has had during your treatment. And again, for likes of audit trails or anything like that, you can follow on, you can see how much chemical you've used. You can compare this with your bug counts, so you can actually see if you were actually testing the bulbs or you were doing any tests on the water. It can automatically be slotted in, so you can actually see the whole efficiency of the system as it's going. Again, we've got 1 or 2 now sort of add-ons which we have produced, which would give you things like gas monitoring. So you've got health and safety issues there which you can control. You can have high-level alarms, so that if there was anything that goes out of specification in terms of your settings, it would have an automatic alert, or indeed an automatic shutdown if that was the way you would wish to set it. We also have the monitoring facilities. This is part of our computerized system. Up here at the headquarters here in Kelso, we have now a team of 3 people whose job it is to basically control and monitor the efficiencies of the dosing systems. So in actual fact, we could actually take control of the dosing for you if that was a parameter that any particular company was interested in. And this is probably more as a general package for a multi-purpose farm. In other words, not just on the bulb washing, but into vegetable washing or into irrigation or glasshouse type treatments. We have now also ventured in for the future. We're also using this technology now, looking at things like control of aspergillus in chitted barley. We've been looking for, in fact, one of our friends who's also a bulb grower, who's been looking at shelf life extension of tulips, and we've also been looking at nematode reduction in your bulbs, because we were very interested to find out how deep we could actually penetrate with the chlorine dioxide into the bulbs. And if you can see here, we've used a blue tracer dye, and you can see that we've actually managed to successfully get the chlorine dioxide into the bulb, and therefore we have implications as possibly being able to control nematodes. So these are projects which we'll be looking at as an ongoing basis, along with the continuation of the fusarium work. So thank you for that. I kind of left it kind of open because I think the questions are probably more important than maybe what I've shown you now, because many of you have already seen some of these results, but hopefully some questions can come along and we'll be able to go into more depth into it.
Thank you very much, Ewan. I think that was really comprehensive. Got a couple of questions. Again, if you do want to submit a question, please do so via the questions box on the panel. So you're saying about some of the trial work that you've been doing on the control of nematodes. So at this stage, you don't have any definitive results? So that's in process, is it?
That's in process. It's something which we started last year, and last year we basically kindly got some sacks of bulbs from Julian, and we've basically been looking. We bought ourselves a tank. So we now have a tank where we can control the temperatures, and we were doing basically time tests. So I think if we look at that one there, you'll see some of the times we've been looking at 45 degrees for 1 hour. We've been looking at 60 degrees for 30 minutes. We've been looking at different aspects of it, just to try and see whether or not temperature made a major difference. And then we were also looking at, and I think it's probably relevant to the question which Rob was given in terms of cold water. So we've also had a look at whether or not reducing the temperature, in other words, could we get away of saying we don't need 45 degrees, we might only need maybe, maybe if we could do it at 10 or 15 degrees or even possibly lower, would we get the same effect in terms of penetration? And that's one that we've just completed, probably, I think it was maybe 2 months ago. And we found now that the difference between temperature is not as significant as we would have thought. It's actually more or less the length of time. If we go down to 10 degrees, for example, we can get the same results, but we might have to go for a dip of maybe round about an hour more than we would in terms of this particular one here. So we're an hour longer, but we've got 30 degrees lower water temperature, and I would assume that the economics of that would be in the heating oil.
Yeah, that's interesting. I'm sure that there would be a lot of growers interested in seeing and hearing about those results as well going forward. Inevitably, the question of cost comes up. Can you estimate the cost of system installation and management with that commercial setup for Narcissus?
Yes, I can.
It's going to be a bit variable, isn't it?
No, I've actually, because we've now been able to finalise the sort of design of the equipment, we can give you almost a definitive type of price range on it. Now, the units which we're going to produce what you might say a catalogue, and we'll have all the specifications, and a lot of them I've made as variable so that you can either choose to have them or not have them, and it won't affect the performance of the chlorine dioxide per se, but it might mean there's a little bit more manual intervention taking place. Now that small unit which we looked at the very end, which was called the Alpha R, that's coming in at probably, no, it's probably coming in at installed with its various activation chambers and the bits and pieces we need. It'll probably be coming in at just less than £1,000 for that one. So that's okay. That one there. The other one, which is the main monitoring system, which is the all singing, dancing type of unit, which will automatically control the dose rate, automatically monitor and record the residual values, and obviously give you control over multiple tanks. That they would be looking at round about £8,000. We can take some of our units all the way up to in excess of £12,000 if you wanted gas monitors onto it, and health and safety features like locking it all off in terms of tamper proofing and other things. But they would be sort of a discussion on an individual company by company basis, and it would also depend. As you see, Julian's one is outside, well out of the way of anybody else. The need for those type of systems might not be the same for him as it might be for some other people who have got maybe a high traffic of personnel and staff around the areas. But that gives you a rough idea. I think the main cost really in all of this is going to be the chemical running costs. And we did say that when we did look at this, we were talking in terms of probably around about, I think it was £8 per litre we were looking at. And we've now got that down significantly. It's not quite half that, but it'll be getting on towards that, because we've now, as I say, commissioned our own production facility for the sodium chloride, which is one of the main precursor ingredients you need for making chlorine dioxide. That we used to have to purchase, and then we would have to then blend it down in our own unique way, because our chlorine dioxide is not the same as anybody else's. We've actually got a very low alkali system. So yes, the costs are coming down. I would reckon you need probably about 6 litres of chlorine dioxide now at, let's call it £5 a litre. You would need. Most of the initial part of it is actually in getting the residual value up to that point of 5 part per million, and then after that it basically just sits there by adding mils of chemical per 20 minutes. So the main hit, you probably have to use you might have to use 2 to 3 litres in the first 30 to 40 minutes, and then the rest of it tails off over the 180 minutes. So it's probably in around that area.
Great, thank you very much. I think it's useful to know that there's some flexibility around the potential options for installing this equipment. I'm going to move on to the last part for now, and again, if there are any further questions, just keep putting them in the box, and then I'll try and come to a couple at the end. Thank you very much, Ewan. I hope you can all see my screen okay. Okay, so I just wanted to share a couple more slides with you. We asked the growers who were involved with trialling the application of chlorine dioxide to their water treatments a few questions during that process. And I just wanted to share those comments with you. So the general motivation for participation was the potential for technology to significantly reduce the levels of fusarium in bulbs, unsurprisingly, which of course could have potential economic benefits, again, depending on the other costs involved with the setup. Also, the associated improvement in bulb health was high on the agenda, alongside hopes for reduction in energy use when carrying out hot water treatment, if it reduces heat use levels and also time taken potentially. Even as a prototype, the growers thought the system was easy to use, but would require commercial adaptation for Narcissus water treatment setups. As you can see from the comments here, at the time of these questions, there were still some unknowns about the technology that could be made commercially applicable, which Ewan has helped clarify in his talk, and has been working with his team to produce suitable applications. So that's encouraging steps forward. In general, the perception by growers involved has been positive, and there is the perception that there is clear commercial relevance. And with the consideration of the results shared by Rob on the efficacy of the treatment in reducing fusarium, growers may well look to trial the commercial application of the technology as a next step. We will be building the information that was presented in this webinar onto a web page on AHDB website so that you can access that and find more information. But in the meantime, please do get in touch with us if you're interested or have any further questions on this. So with that, I will quickly come back to the questions to see if we have anything further. Let's have a quick look. I think it seems like we've got some satisfied audience members because there's nothing else coming through at the moment. As I said previously, if you do have any further questions once we've finished, please do just drop me an email and we'll follow up with you. Thank you very much to Rob and Ewan for their presentations. Really appreciate you doing this with us. To remind you, the webinar recording will be made available on the AHDB Horticulture Events archive page. So if you do want to catch up or refer anyone, that will be there to access shortly, alongside the Narcissus crop protection webinar, which we held on Tuesday. So both of those will be up there, hopefully in the next couple of weeks. Otherwise, don't forget your points. Make sure that you send those forms to Maya, and if you have any issues with that, again, just let me know. But otherwise, thank you very much for listening. I hope you have a good afternoon.
Thank you very much, everybody.
Thank you.